Hello, this is Mrs. White, and I'll be talking about mutations. So we've been talking about mutations since last week regarding about the change in the gene of organisms. And so these gene changes may be helpful, harmful, or no effect to the survival of organisms. Now, just to recall, what are mutations? Mutations are changes in nucleotide sequence of DNA and may occur in either body cells or called somatic cells, or may occur in the gamete. Normally, they are, of course, in egg and sperm cells and can be passed on to the offspring. If it is a somatic or body cells, normally it doesn't need to be passed on, or it cannot be passed on to their offspring. Now, are mutations helpful or harmful? Now, just to keep in mind, mutations regularly happen, even if we don't know it. Some mutations are neutral, meaning it doesn't have any effect on humans. Now, some chemicals or UV or ultraviolet radiation may cause mutations, and these many mutations are repaired by enzymes, so therefore it may not affect the organism. Now, some types of skin cancer, for example, and leukemia, are a result of somatic mutations or body cells mutations. And some of the mutation may improve organisms' survival, which is beneficial for the organisms. Otherwise, it may harm the organism. Now, let's take a look at the types of mutations. Now, the first thing that I'd like you to remember is that mutation can be a chromosome mutation. Now, this may involve the changing of the structure of the chromosome. As you can see here, our chromosome structure. It can be a segment lost or a segment gain on the part of our chromosome structure. Now, there are 5 types that exist. First, we have deletion, then inversion, translocation, nondisjunction, and duplication. We're going to take a look at each of them. We'll start first with deletion. Now, deletion is the result of a breakage in the chromosome. And a piece of the chromosome is normally lost. So if you look at this diagram right here, the original chromosome is the left-hand side, and a portion of the chromosome breaks off or get deleted, and therefore the chromosome is shorter than usual. An example of that is some physical deformity like this webbed hands or other deformities in the fingers or digits of an organism. Next up is inversion. Now, inversion happens when chromosome segments break off, and after it breaks off, it actually flips around backwards, and it reattaches itself in that same segment. So here's an example of how it looks like. So this breakage doesn't necessarily comes off. Unlike deletion, it really comes off. In this case, it moves and flips backwards, and therefore it causes inversion. So an example of an inversion mutation is what we call hemophilia. Hemophilia is a chromosome mutation, happens in the X chromosome, which is either X chromosome in the gender, X and Y chromosome, X and X chromosome. You guys will learn that in high school. And it causes non-stop bleeding on the person who has this chromosome mutation. The next one is your duplication. Now, duplication occurs when a gene sequence is repeated, meaning there's like another copy of that same segment. So if you look at this diagram right here, the original chromosome has certain bands of yellow, and if there is duplication happening, it's another section that's copied or duplicated. An example of a disease that is caused by this mutation is your Charcot-Marie tooth disease. Now, it doesn't have to do with the tooth, but it causes a high arch in the foot of the person, and most of the time this mutation causes the person to not be able to walk. The next one is translocation. Now, translocation involves two chromosomes that aren't homologous. Now, part of the chromosome is transferred to another chromosome. Now, your chromosome structure, like the whole entire genome of a person, there are 23 pairs of chromosome. Now, in this example that I have, which is Down syndrome, there is a specific chromosome number, or the 21st chromosome. Instead of having 2, it has 3 chromosome in it. That causes the Down syndrome. We also call this disorder trisomy 21. The next one is nondisjunction. The nondisjunction is the failure of your chromosome to separate during meiosis. And this causes the gametes to have too many or too few chromosomes. So in this example that I have, which is Turner syndrome, instead of having 46 chromosomes, there's only 45 because it is missing one chromosome, especially on the sex chromosome, which is the very last pair of chromosomes. So your last chromosome is actually your gender chromosome, either XY for males, XX for female. And in this particular disorder, this person is missing another X chromosome. So this is a female, and this female is missing a chromosome, and therefore it causes a Turner syndrome. And the Turner syndromes have some effects on a person. An example would be low hairline, they have heart problems, and some of them are not able to have children, and as well as having webbed neck. And these are some of the physical deformities for nondisjunction. Now, the nondisjunction, like I said, normally happen in meiosis, so this is an example of a meiosis. I'm not going to get into the detail, but this causes, kind of like a missing chromosome in the genome of humans. Now, this is the last part before I summarize the chromosome mutation. So this is just a little animation of each type of mutation. So in this case, that's translocation. This is duplication. Now this is the normal chromosome, and once it's deleted, it's shorter. And this is your inversion. Last but not the least, your translocation. I hope you understand and probably have some idea of the different chromosome mutations that we discussed today. And I'll see you in class.